Hi students, I'm Dr. Anandhi. Welcome to my channel Make Biology Simple. In today's session, we are going to see the BDS lecture 3 for the first year BDS students, carbohydrates. In the carbohydrates, we are going to see different type of structures, how to write the basic structure, and how the bonds are involved for joining the more of one or two sugars to frame the disaccharide as well as polysaccharide. So totally today's session is about the how to write a structure of the glucose or the fructose, or how these carbohydrates are joined together with the help of the glycosidic bonds, and what are the other bonds involved for the bonding. All these details we are going to see in the today's video. So you can write the structure of the glucose in any of the form. This is, for example, this is the structure of the glucose. Since the formula of the glucose is C6H12O6, you can write this. This is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. This type of writing the structure of a glucose is called linear structure. So same way, this is called Fischer structure. We have three different structures for any monosaccharides. So first one is a linear structure, next one is Fischer structure, third one is Howard structure. So one by one we'll see in the slide. So this is an example for the glucose. Suppose if you are going to write the Fischer structure, here you can see the intra bonding. So intra means within the structure you are getting a cyclerized. So which carbon is involved in the cyclization? Well, we'll see for one. First one is the first carbon and the fifth carbon. The fifth carbon we have an hydroxyl group, and in the first carbon we have the methyl group, aldehyde group CHO. So what happened here? A common hydroxyl oxygen is combined, and a hydroxyl group, see this OH is removed, so OH is converted like this, and in the O you have a oxygen bond which connects the C1 and C5. This is structure called intra bonding or intra oxygen cycling process of the glucose molecule. So that is called Fischer. And why? And this structure we otherwise call alpha D glucopyranose structure. Glucopyranose structure. So this is how you can write the Fischer structure. The next one is the Howard structure. So from the Fischer you can write the pyranose structure. So this is a six membrane ring structure. This is six membrane ring structure, and here starts the first carbon. So let it be, this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. So numbering is very important. When you know the number, you can write the structure very easily. The first, second, third, fourth, fifth, sixth. So this is the sixth CH2OH. Now fifth, you can see that COH, and this is bound to the first carbon. See, first carbon and fifth carbon are intra bonding is formed. So here C5 and C1 is involved in the intra bonding, and then fourth carbon CHOH is written. So OH is C, and then this is your third say third carbon, and here's the second carbon. This is the first carbon. So 1, 2, 3, 4, 5, and 6. So this is the numbering of the carbon. So you can see this 5 is CO, CO, C, OH, and this is HOH, and you can see the difference OH in the left side. So that's why the OH is been added in the upper portion. When it is lower means it comes to the left side. So this is how you have to write the structure of the glucopyranose structure. So that is otherwise called Howard structure. Next was fructose. Fructose, it's a keto sugar. So in the first and the sixth carbon you have OH group. Second carbon you can expect a ketone group. Then OH, here OH and OH. So this is the structure of the fructose. The fructose can be written as a Fischer structure like this. Since here the second carbon is having a functional group, here instead of 1 and 5, here 2 and 5 is intra bonding is formed. So here in the OH group, the H is added to the here CO H, and one more H is attached. So that is how the intra bonding is formed here. And then this is a structure of a Fischer type for the fructose. Next comes the furanose structure. This is um, a ring structure. Here the ring is having the furanose type. Furanose, this is if you have the hexagon ring, it is called pyranose. If you have this 5 membrane ring, we call it as a furanose structure. So furanose structure numbering, you can see how the carbons are attached: first, second, third, fourth, fifth, sixth. In first and the fifth, last carbon, you have hydroxyl group. So here it started the first carbon. This is 1, this is 2, this is 3, this is 4, this is 5, and this is 6. This is how the 6 carbons have been attached in the furanose structure. Now this structure we call it as a Howard structure. So for any monosaccharide you can write a linear structure, closed one is called Fischer structure, and ring is called Howard structure. So these are the structure how to we mentioned how to frame or draw the structure for the monosaccharides. Now we are moving to the isomerism. So what is isomerism? Isomerism means a compound possessing same molecular formula but differ in their structures. Differ in the structures are called isomers. So this isomer is called, the phenomenon we call it as isomerism. So two types of isomerism is there. One is structural, another one is stereoisomerism. So structural isomerism is what we are going to see here. Structural isomerism means molecule having the same formula but differ in the structure. So why we are talking about the isomerism means here in the case of carbohydrate, there are two types of structural formation based on the categorization of the carbohydrates. Earlier we have seen aldo sugar, keto sugar. So same here we are going to see the structural isomerism is divided into two forms. One is aldose type, the other one is keto types. Glucose and fructose are isomers because they too commonly sharing the formula C6H12O6, but the structure is totally different. See here, instead of here it's an aldo type, so that's why aldehyde group is there. And in the case of fructose, we can see it's a keto group is there. So aldo sugar is having an aldehyde group. Keto sugar, the aldo sugar example is glucose. Coming to keto sugar, it's a ketone body, and that is called as fructose. So this type is called structural isomerism. Structural isomerism. Next one topic is called epimerism. So we have seen isomerism. What is epimerism? Epimers are the sugars which are differ with each other with respect to a single carbon other than the anomeric carbon. Anomeric is the first carbon which have different thing, and this epimers are the sugars which are differ only in a single carbon. So that is what we are going to see now. Galactose and mannose are the epimers of the glucose. So they differ from the glucose with respect to the C4, C2. Here we are taking the example. For example, we know the glucose structure: CHO, CHOH, COHH, HOH. Here only in the third carbon, the OH position is in this side. But coming to the epimers, you can see the same structure here. If you have the second carbon also, the OH to the right side, then it is called mannose. If the glucose structure same, when you compare here, if the fourth carbon also is having this OH group this side, that means the left side, then that is called galactose. So these are called the structures of the epimers for the glucose, galactose, and the mannose structure. Nothing but epimers also belong to sugar type, but they differ in each other with respect to a single carbon. That means here you can see in this case the fourth carbon is changed, here second carbon is changed with respect to the common structure glucose. So if these two sugars are changes, or these two carbons are changes, that type is called epimers. Now the next thing is, based on the hydroxyl group's attachment, we call it as two different glycosidic linkage formations are there. So one is called O glycosidic linkage, the other one is called uh, N glycosidic linkage. Why we call N and O, we'll see now. So any carbohydrates, when they come close together, we have seen already examples, the Haworth's projection formula, Haworth's projection of writing the glucose molecules. This is a Haworth's projection of two glucose. So when they two combine, we called as a disaccharide. When if it more than two is formed, we call it as oligosaccharide. If you have n number of glucose units joined together, we call it as polysaccharide. This is what we have seen under the classification. Say for example, here we are talking about the glycosidic form. formation. So glycosidic is nothing but the inter bond formation. We have already seen in the Fischer intra bonding formation. Here we are going to see the inter. Inter means between the two sugars, how they are linked together. So you know this is the first carbon, this is the fourth carbon. This is the upcoming sugar, this is the existing sugar. So the first carbon and the fourth carbon hydroxyl group. Here one OH, here one OH will be there. If they two combine, a molecule of water is removed and they are linked with the O group. That is O nothing, oxygen of the existing and the upcoming glucose molecule. Now this structure we call it as a glycosidic bond. So, two molecules of glucose join together with the help of the glycosidic bond. Since the oxygen is involved in connecting, that is called O-type glycoside bond. So, this we can see in the example for the disaccharides maltose. So another type we call it as N-type glycoside bond. So N-type glycoside bond is this is an example of your ATP, GTP, all these nucleotides will have. Here what happened? This is a ribose. Ribose we know it is a pentose sugar, 5-carbon sugar. There in the first carbon, it is involved with the base of, what is this? This is the adenine. What is adenine? Adenine is a nucleotide base. So the nitrogen is getting attached with the carbon of the ribose sugar. What is ribose? Ribose is a penta sugar. This is also coming under the carbohydrates. So the ribose is the first carbon. It is attached with the adenine, that too in the nitrogen portion of the skeletal of the adenine. So those type of structure is called as N-type glycoside bond. So there are two types of bonding formation. One is O. When oxygen is involved, we call it as glycoside, O-type glycoside. When nitrogen is there involved in the formation of bond, then we call it as a N-type glycosidic bond. So I think this session today we have seen only the bond and how you have to write the structure in different ways for the glucose and fructose. So one is a linear, second one is a Fischer projection formula, and third one is Howard's projection. And then if these two glucose structure or any carbohydrates monosaccharide combine, they can form a glycosidic bond. So one is O side, that is oxygen bonding, and the other one is nitrogen. These two type of bondings are generally we can able to see in the with what is called in the carbohydrates chapter. So where does these bonds, the glycosides are important? So these glycosides are mostly involved in the drug structure, which is used for the treatment of tuberculosis. And one more glycosides we can see, it is cardiac glycosides like digoxin, digitoxin, or these are the muscle contractions thing, and they are used for the treatment of congestive heart failure. And one more glycosides we are going to see is anthracycline and glycoside like used to treat the wide range of cancers. One more thing is quabin inhibitors of the sodium potassium ATPase and block the active transport of sodium. So these are the four different physiologically important glycosides: streptomycin, cardiac glycosides, anthracyclines, glycosides, coabin. So these are the four different. Generally, in our day-to-day use, we can see these type of drugs are having this glycosides. What is glycosides? We have seen on the glycosidic bonds, no. So the compounds which have the glycosidic bonds that referred as glycosides, and the general physiologically important glycosides we have seen in this session. So once again, I conclude that we have seen about different types of writing the structure of glucose in the form of linear, then Fischer, then Howard's projection, and then how they are linked together with the help of two different bonds. One is O, another was N, and the physiological important glycosides. I hope this video will be very useful for the basic fundamental understanding of the carbohydrates, of about how to write a structure and the bond involved in the carbohydrate linkage. That's all in today's video. Thank you, students.